Hello, my name is Dr. Jordan Smith. I'm one of the pharmacy faculty at the Fred Wilson School of Pharmacy at High Point University. And it's my pleasure to be able to talk to you all about some of the interesting and important work that's going on in the world of vaccine research, specifically pertaining to COVID-19. Hello, everyone. Today we're going to be talking about the currently available COVID-19 vaccines. We have a couple different types available. One of them, the same technology at least, provided by Moderna and Pfizer. Those have been around for a little bit longer. And then, of course, the Johnson & Johnson vaccine, the one-dose vaccine. I'm going to be breaking down sort of how both of them work. We're going to start with the Moderna and Pfizer vaccines and then make our way down to the Johnson & Johnson. So to begin, Moderna and Pfizer built their vaccines on mRNA technology. This is brand new and it's incredibly exciting technology. These are the first vaccines that have ever been approved, emergency use authorized using this technology. And I want to make a quick point that it's not just for COVID-19. The success of this type of vaccine has made research into HIV, oncology, all these areas just reinvigorated. And I'm excited to see where we kind of go from here with other mRNA vaccines. But we're talking about COVID today. So mRNA codes for proteins. Well, the COVID-19 virus, pictured here, looking real sad, has lots of proteins. The protein of interest for us is one of the surface proteins known as the S protein. You can see it here and here. Basically sticks off that COVID-19 molecule. Why is that S protein important? Well, one of the big reasons it's important is our body's immune system recognizes that S protein. And it's one of the primary targets that our immune system finds and attacks when COVID-19 is replicating and causing damage. Now, because we're so on the ball with learning about COVID-19 and understanding that genome, we were able to take that S protein, learn the code, and then get the mRNA that codes for the S protein out of that. And then lo and behold, we've created a vaccine out of that mRNA molecule. So the vaccine itself is the mRNA that codes for, is translated into that S protein, wrapped up inside a lipid nanoparticle. This lipid nanoparticle keeps it stable, keeps it safe for storage and for entry into the host cell. Now, once that mRNA vaccine is ready, it's prepared, it is injected into a person, you or me, I've had two of them, it hits the host cell. The mRNA is transmitted into the cell, so this envelope kind of binds with the cell, buds onto it, and then the mRNA is released into it. And now, here's where things get a little bit different between how the two vaccines work within the cell. First and foremost, I want to say, the mRNA vaccines never go anywhere near your nucleus. They don't get incorporated into the DNA in any way, shape, or form. They kind of swim around in the cytoplasm and work their way toward the endoplasmic reticulum, where we have ribosomes. So DNA hanging out in the nucleus, minding its own business. For us, and what we're interested in, is mRNA comes in and uses our ribosomes to create this S protein. So translation from the mRNA into a protein with the host ribosomes. Now we've got our protein intracellular, and that protein is going to travel its way out to the edge of the cell and get expressed. There's a couple of places on the outside of cells that the protein can get shown and expressed to other cells within the body. They're going to help us fight against it. One of them is an MHC2 receptor. The other is an MHC1. We're going to break down what the two of those mean and how they're important. All right, so MHC2 cells are present on only a certain number of cells within the body. These are what are known as antigen-presenting cells. So not every cell is capable of presenting MHC2. MHC2 is important because these antigen-presenting cells, things like macrophages, dendritic cells, they present that protein and that alerts our body's immune system, specifically in the form of T helper cells. These T helper cells, otherwise known as CD4 cells, are capable of recognizing that protein on an MHC2. When they recognize that protein, they say, oh gosh, there's something here, I need to take care of it. And they go ahead and start going down all sorts of different pathways. These T helper cells produce little interleukins. These interleukins are molecules that circulate throughout the body that send signals to other cells and back to themselves as well to continue the inflammation process, continue immune memory, all sorts of fun stuff. Specifically with the T helper cells, we see interleukin 2, interleukin 4, interleukin 5. What do these interleukins do? Well, they come out here and they find the B cells. So we've got T cells, we've got B cells. Each of them is integral to forming a complete immune response. Once the B cell recognizes all of these interleukins, once it recognizes the help from the T helper cells, right? T helper for help, it differentiates into what are known as plasma cells. When the B cell differentiates into a plasma cell, these plasma cells secrete antibodies. Antibodies, we've heard that term so often over the last year. Well, these antibodies are what we need to fight off that COVID molecule. So what we've managed to do is go from this MHC2 presentation of that protein, the T helper cell recognizing it, that T helper cell signaling to our B cells, hey, it's time to mature into a plasma cell, those plasma cells producing antibodies, those antibodies going out to target COVID-19. You can see these antibodies here latching onto and attacking those S proteins on the outside of the COVID molecule. One other important function of the T helper cells, again, signaled by MHC2, is that they can differentiate into memory T helper cells. So we don't only get memory B cells, we also get memory T cells. And these are going to hang out for us for a long time, and they're a cornerstone of what provides long-term immunity to the virus. So going back to our cell here and that MHC1 receptor, MHC1 is on every cell in the body. MHC1 presents foreign material and signals out to our immune system, hey, there's damage here. You need to take care of this. Essentially, it signals that it's time for that cell to be killed. So this MHC1 receptor presents the protein to CD8 T cells. These CD8 T cells are what are known as cytotoxic T cells. That's the TC. The cytotoxic T cells are programmed to recognize foreign protein on MHC1 and then start the cellular destruction process. So we can see here that we have an MHC2 based immunity where the MHC2 signals to T helper cells and B cells. The T helper cells help the B cells differentiate and create antibodies down the line. The MHC1 molecules signal to CD8 cytotoxic T cells. These cytotoxic T cells are there if we have enough damaged cells and they need to be destroyed. You can see this red X for destruction here. One important thing I want to point out as well, though, with the vaccine is that a T cytotoxic cell is not going to immediately destroy a vaccinated cell. At this point of our immune function, we are recognizing and creating that cytotoxic T cell response. So in the future, when this protein is expressed on the surface of a cell, then we're going to start to see cytotoxic responses. So overall, we have mRNA being injected into the person, going into that cell. That mRNA is translated into a protein. That protein gets expressed on the external part of the cell in a couple different places. We either have our T helper cells that help our B cells that eventually create antibodies against that COVID-19. We have our MHC1, our cytotoxic T cells that will take care of any cells that are infected down the line. Each of these is incredibly important to the overall immune response, and each of them have extraordinarily good response to vaccination with the mRNA vaccine. So now I want to shift from the mRNA vaccines and talk a little bit about the Johnson & Johnson vaccine that is built with an adenovirus. It is a DNA vaccine housed inside an adenovirus. Now, that sounds kind of interesting, right? We've got a virus we're trying to vaccinate against. Why are we using another virus to introduce it to the host? Well, this adenovirus that's used in the Johnson & Johnson vaccine is not pathogenic, meaning it doesn't cause illness. It's one that our bodies are able to recognize and incorporate without causing any issues. The important piece of this vaccine is, of course, the adenovirus capsule, but then the DNA that was created from that same S protein that was used for the mRNA vaccine. So we've added in one extra step. And in this case, rather than like with the mRNA vaccine going from protein, mRNA, get that into the cell, we've gone protein, mRNA, went ahead and taken another step, made that back into DNA, and then that DNA is inside our adenovirus. This matters in terms of how the vaccine initially handles itself, but we're going to see that the end result is largely the same. So with the Johnson & Johnson vaccine, we've got our DNA encapsulated inside that adenovirus. That adenovirus is injected, and adenoviruses interact with the cell a little bit differently than a lipid nanoparticle would, but essentially gets into the cell. The primary difference here, and the one that's important to realize, is that here we do go into the nucleus. We do use nuclear machinery of the host to ultimately end up with the protein that we want to express. So once our Johnson & Johnson vaccine is injected, that DNA from inside the adenovirus gets in here, gets into the host nucleus, and never interacts with our own DNA. And I want to make that point as well, because gosh, this one sounds different. It gets into the nucleus. Could that be a problem? It is not something that intercalates into our own. It acts independently and again uses that cellular machinery, sort of like the mRNA vaccine does. So once the DNA is present within the nucleus, that DNA uses our own cells molecules to create mRNA. That mRNA looks a lot like the mRNA we had in the other vaccine. It is pushed out of the nucleus. The mRNA encounters the ribosome. At this point, the response is largely identical to what happens with the mRNA vaccine. The ribosomes translate the mRNA. The mRNA produces that protein after translation. This protein gets expressed at the cell surface just like it did in the mRNA vaccine. So the whole cycle repeats itself from this point forward. And just to reiterate, you've got your protein being expressed. That protein goes out to MHC2 and MHC1. MHC2 is recognized by T helper cells. Those T helper cells help activate and differentiate B cells. They also produce memory T helper cells. Those B cells mature into plasma cells. They create the antibodies. The antibodies find the COVID swimming around in the bloodstream and the rest of the person's body, and they attack it. We also have recognition by MHC1. Cytotoxic T cells recognize MHC1. These cytotoxic T cells learn to recognize that specific protein, and then in the future, when MHC1 is presenting the viral protein, they attack the infected cells. This happens in both cases, Johnson & Johnson and Moderna. Really, the only differences are Johnson & Johnson is approved for 1 dose, Moderna and Pfizer are approved for 2 doses, and of course, how the initial portion of this immunization occurs. Do we go straight to the mRNA, or do we start at DNA that needs to be transcribed into mRNA and then ultimately translated into protein? Very similar situations, incredibly effective vaccines, extremely grateful we have both of them, and happy to have been able to spend a little bit of time talking to you about them today. I want to thank you all for taking the time to watch me try to explain the COVID-19 vaccines as they're currently available. And I hope that our time today has helped you understand a little bit better how these vaccines work. And most importantly, get vaccinated, take care of yourselves, encourage your friends, your family, everyone to get vaccinated. We're in this together. Let's get it done. We can see the light at the end of the tunnel. Thanks again for your time, and enjoy your summer.